All right, before we start, I am going to pretty much put forward a disclaimer on the videos and the PowerPoints that I'm about to show you. Some portions of this, the video portions, which are halfway into the presentation, contain some graphic scenes. It's going to contain scenes of me cutting into a not live but a real mouse that has been euthanized, and it could be a little bit graphic for some of the girls or some of the guys in the class, so you don't have to watch it if you don't want to. I realized that I didn't really have an opportunity while I was in class to talk much about the research that I do. It's actually an extremely interesting field of research. I work with neurodegenerative diseases. So these are diseases that cause the destruction of the nervous system. And what I'll do is, or what we do as scientists, is we take these diseases that occur in humans. So this might occur in, for example, my grandma had one of these diseases. So we take these diseases that occur in humans and we recreate them in mice. This allows us a simple organism to study these diseases. As part of this study, what we will do is we will take the spinal cord and the nerves, which are two components of the nervous system, from these mice, and we will use those in our studies to classify what is going on in the disease. If we know what's going on in the disease, then we have a better artillery or a better means in order to help those who are afflicted with the disease become better or reverse some of these signs of the disease that we see. Now let's talk about the cardiovascular system. The cardiovascular system has three main parts: the centralized heart, and then the two disseminating parts, which are the arteries and the veins. The arteries are the parts that carry blood away from the heart, and the veins are the components that carry blood to the heart. At this point in time, Miss Parker should be passing out pieces of paper that have an area for responses to 6 true and false questions. So I'll give them some time to hand these out. And while they're handing these out, I will give you the questions that you guys are going to be answering. You'll use the diagram in front of you, and I'll leave this picture up throughout the duration of this short little quiz, but each group of questions is going to pertain to a particular region on this diagram. For example, the first two questions are going to revolve around this blue structure to the top left of the heart, where the next two questions, 3 and 4, are going to pertain to the red structure to the top right of the heart, and the final two questions, 5 and 6, are going to pertain to the structure to the bottom left of the heart. Alright, now I will wave goodbye to everybody and give you 5 minutes to answer the questions. Again, the answers are going to be either true or false, and then we will review the answers whenever I come back in that 5 minutes. Mrs. Ford and Miss Parker, make sure that they're staying dedicated and diligent to their work. Alright, see you guys in 5 minutes. Alright, now remember that the first two questions revolved around this structure in the top left corner of the screen just above the heart. It's this blue circular looking structure. And the first question reads, the first statement reads, blood from the legs enters the heart here. Is this true or false? I will tell you right now, I don't want to give all the answers away because if I click the button now, all the answers will come up, but this is actually false. Blood does enter the heart through this segment, but since it is coming from the upper portion of the body, it is not blood from the legs that actually enters here. And since blood is entering the heart, blood always enters the heart, or most of the time enters the heart through veins. So this structure is actually a vein. Next two questions, questions 3 and 4, or so let me go back. So 1 would be true, or 1 would be false, and 2 would be true. The next two questions, 3 and 4, center around this structure, the red structure to the top right of the heart. And the first question says, the first statement says, blood leaves the heart here. And since it's the right portion of the heart, blood actually does leave the heart here. And the question number 4 is pretty much asking if blood leaves the heart from veins. And since we've already seen that blood enters the heart through veins, it's most likely that the same structure is not going to cause blood to leave the heart. So the number 4 is actually false. This structure is actually called an artery. Anytime blood is going away from the heart, it's going to be through an artery. The final two questions, 5 and 6, center around this blue structure to the bottom left of the heart. And blood, the first statement reads, blood from the legs enters the heart here. So this structure is actually coming from the bottom portion of our body, the lower portion of our body, so blood from the legs is most likely being carried inside this structure. And similarly to what we saw above, where blood was entering the heart through this vein, the statement number 6 says this is a vein, which is actually true as well, because blood is always entering the heart through veins. So as a wrap-up, 1 is false, 2 is true, 3 is true, 4 is false, 5 is true, and 6 is true. Next, you notice that whenever we were talking about the heart, it was separated into both left and right sides. The interesting thing about the heart is that the sides are determined by the orientation the heart takes within the body, not the way you look at it. So if you're looking at me, the right side of my heart would be on my right side, and the left side of my heart would be on my left side. So whenever you're looking at it, it looks like the left side is actually on the right to you, and it looks like the right side is actually on the left to you, which is what we're seeing whenever we look at this screen. If you look at this point at the bottom portion of the heart, that is actually the line that is going to separate both the left and right side of the heart if you draw a line all the way up. But whenever we flip over the right side of the heart to the left side of the heart, what we notice is that one of the structures is larger on one side than the other. There are some similarities between the left and right side of the heart, where both the left and right side have an upper structure and a structure on the top where the blood will enter. These are called the atrium. Next, we have a lower structure that the blood will pass through after it enters each atrium, and these are called the ventricles. So these are similar structures between the left and right side of the heart. But whenever we start to get into some unique structures between differentiating the left and right side of the heart, we're talking about the superior inferior vena cava. Vena indicates it's a vein, and therefore we can conclude that blood is entering the heart through the right side. And if we notice in our diagram, the right side, this atrium, actually is larger, and maybe that's because all the blood from our body is entering through this point. Next, blood is passed to the lungs through the pulmonary artery from the right side of the heart. It will move through the lungs and then leave the lungs through the pulmonary vein, which is a structure that distinguishes the left side of the heart from the right side of the heart. Finally, after it cycles through the atrium and the ventricle of the left side of the heart, blood is pumped out through the aorta. And the aorta is actually an artery. So blood enters the heart through veins, and it will leave the heart through arteries. Now, if you're really smart, there's actually a contradiction to the statement I just made, and kind of think throughout the presentation that I'm giving what might be this contradiction. Finally, let's look at a blood molecule traveling from the legs all the way through the cardiovascular system. So this mesh-like looking thing in the bottom is actually a capillary bed, and we're going to consider these capillary beds where the legs are. So if there's blood within these capillary beds, then they're within the legs. So blood, the blood molecule, a single blood molecule is in the legs, and it is going to travel up to the heart through a vein until it reaches the inferior vena cava, because it's coming from the legs, and blood from the legs enters the inferior vena cava and will enter the right side of the heart through the right atrium. It will then move to the right ventricle. From there, it will move up through the pulmonary artery into the lungs. and then be transported after it's oxygenated through the lungs into the pulmonary vein. So the pulmonary vein will then bring it into the left atrium, where it will then move to the left ventricle, and from the left ventricle it will exit the heart through the aorta. Finally, whenever it gets carried back to the legs in its oxygenated form, it will be carried in arteries. So now let's move from the diagram of a single blood molecule moving through the circulatory system and talk a little bit about, or show you a little bit of the real thing. In this case, whenever we are perfusing these animals, they're going to follow the same path. The solution is going to follow the same path as the blood molecule that I showed to you guys earlier. So be thinking about the path of blood and the path of the solution through the circulatory system, through the cardiovascular system, as this experiment, as the movies are playing. It will help you understand it a little bit later. The first thing we need to do is, you will see this chemical in the background called isoflurane, and this is actually an inhaled anesthetic. So what this is going to do is, it's essentially going to knock the animal out so that they can't feel any pain. Every single time we conduct an experiment like this, it needs to be done in a manner in which the animal cannot feel any pain, and this is done by using isoflurane in this case. What we will do is we will administer a small dose of this, and we're going to regulate this by pouring it out of the main stock container into an individual tube that I can control the amount of isoflurane that the animal will inhale. So it's kind of a rough estimate, but I just pour probably about 2 or 3 drops into the 50 ml conical tube, which you see in front of me, and I'm screwing in, and then make sure that everything is closed back up and in its proper place before I proceed. Next, I take the animal and I put his face pretty much into the tube so that none of the anesthetic can leak out, and that he will be efficiently sedated. Next, what I'll do is I'll monitor his breathing habits. What you'll notice on this close-in view is he has very quick breathing at this point in time. When you know that he's completely knocked out, his breathing will start to slow down until the point where he'll start to have like large gasps. Then he'll have a final gasp, and that will be the point in time in which he is completely anesthetized and it is fine to continue on with the procedure. So this is the anesthetized animal. The first thing we need to do to this animal is, since we're going to dive in and be looking for organs, we need to make sure that he's stabilized. So we're going to stabilize him with these surgical pins. These are going to go through the armpits and the back of the legs, so the armpit of the leg as well, of each leg and each arm. Hopefully I'm not blocking your view with my hands. This is one of my first times using a camera and also doing this experimentation, so what I try to do is keep my hands out of the way as much as possible, but there's going to be times where you might not be able to see, but I try to keep that to a minimum. So once this animal is completely spread out and everything is locked in place and easy to see, then we can move on with the procedure. So let's make sure that everything is tight and ready to go, and it looks like it is. The draining of the blood is going to be accomplished by running this solution through the animal's cardiovascular system. It is called PVS. It's pretty much just a salt solution that we are going to run through it in order to push out the blood. What you'll notice is that this is labeled, and then it also has my initials and the date in which this was made, because we don't want to be using an old solution on this animal because that could be bad for it. So let's watch the video and see kind of how the procedure is going to work. The first thing you'll do is unscrew the cap, and there's a hose that leads from the solution to this perfusion machine that we're going to use. the solution will go all the way down this tube, up and around until it gets to a valve. Right now, this valve is set on a red light pretty much. It's a stop sign. Until I twist it over to the right, no solution will be able to get to the machine. So twisting it to the right pretty much acts as a green light and the solution is ready to go to the machine. Then it will go into the machine, it will go around this hinge right here and then come back out and then lead to the animal. We do not want to shoot a bunch of water in this animal, or solution in this animal, we want to be able to control it. So we do this by attaching a syringe and a little bit of hosing to the end of our tubing. At this point in time, the needle is covered because we want to worry about safety because it is very sharp, and I will take that cover off and then we'll be ready to go. Next thing we need to do is before we open the animal up, we need to spray it down with ethanol because this will keep all the hair and everything out of our instruments. It will keep everything a lot cleaner. So here's a close-up view of the incisions that I'm going to be making on this animal. The first thing that needs to be done is you cut the animal right below the diaphragm. First, you need to cut off a little patch of hair and skin so that you can expose the underlying tissue, and then cut through that as well. So we'll speed this up so that you don't have to just sit here watching me cut through this. We'll go 8 times and it should run through this. So now it's slowed down a little bit, and what is actually happening now is I'm exposing the underlying tissue and the liver and all the under organs. I'm cutting up through the ribs to hopefully expose the heart, because at this point in time you cannot see the heart until I cut the ribs away. There you can now start to see the heart, and even though the heart is still beating, that is just a residual action of the animal. And I will show you how I'm pulling the fat tissue away from the heart so that I can get access to the heart, and it will allow the heart to be more visible so that whenever I'm introducing the needle, which needs to go into a specific part of the heart, it's a lot easier to do. Again, sorry for having my hand in front of you, but this was kind of tough to do, so I had to get at any angle that I could. Now the heart is pretty much unsheathed, and what I'm doing is I am plugging the needle into the left ventricle in this case. The connection between the atrium and the periphery of the animal I'm going to sever right now. Then solution is coming all the way out to the animal's body and then coming back in the heart through the right side. But remember, I sever the connection between the right side of the heart and the veins in the animal's periphery. So all the blood that would be entering the right side of the heart is being actually pushed out. Next, we are going to take a little post quiz to, one, assess your understanding of the cardiovascular system as I have detailed it earlier, and second, it's going to test your knowledge or test your understanding of the procedure that we just conducted and see if you can link the procedure to, or see how the procedure utilizes the cardiovascular system, utilizes the basic mechanisms of the cardiovascular system in order to drain the blood. So the first image we have here shows two things. This is our cardiovascular system in the context of the mouse. I'm just using a diagram in order to make it simpler. And we have the needle that is introduced into a certain region of the heart. And then this is the right side of the heart, the connection that I severed whenever you saw me sever that connection. So now what we're going to do is there should be 6 questions in front of you that Mrs. Ford and Ms. Parker are passing out right now. And I'm going to quickly run through all these diagrams so you can see kind of how it works in motion. And then I'm going to give you about 5 minutes to answer all the questions, and then I'll come back and we'll review the answers. So the first question is, what part of the heart is the needle inserted into right now? What we see here is as we start pumping solution which is this blue stuff right here, as we start pumping solution into this side of the heart, blood is going to exit through the exit out of the severed region of the cardiovascular system that I severed. So the next question is, the solution at this point in time is leaving from what part of the heart? So what structure of the heart did I tell you that blood leaves from in the left side of the heart? So then we have even more solution coming through because it's been running longer time, and even more blood coming out. So the solution right now is traveling through a artery or a vein is the next question. So the answer is either going to be artery or vein. Now the blood or the solution has reached the lungs or reached the legs, the lower portion of the body. So if this entire region is filled with solution and blood is coming out, and I said earlier, what did I say was equal to the legs? If this is the case, is there blood in the legs right now is the next question. So this is a yes or no answer. More solution gets pumped in and is further pushing the blood. So now as the blood is traveling back to the heart, is it being carried in an artery or a vein? Finally, since we've severed this connection, once the solution gets here, is it going to enter back in the heart through the right side, or is it just going to go out to where the blood is? So now I am going to give you probably about 5 minutes to answer these questions, and then we will go back through and review all the answers. Next, we are going to take